we are thrilled to present new immune function tests to assess immune age, systematic chronic inflammation, and design therapy, brought to you by integrativepractitioner.com and sponsored by Cell Science System. I'd like to welcome our speaker for today. We have Roger Deutsch, who's the founder and CEO of Cell Science Systems. And at this time, I'd like to hand over the session to Roger. Welcome, Roger.
Hi there. Thanks very much. Appreciate that, Laura. Thanks everybody for taking time to attend. What we're going to talk about today are some new tests that we've developed that are useful for test assessment of immune status, health, and measurement of chronic inflammation. And we think it's a particularly important time in history to be looking at these. If you're not familiar with Cell Science Systems, we've been around for a little more than 20 years, and we are always involved in a lot of different research projects. These are some of the collaborations that we've had over the last few years. You'll see that we're a CLIA licensed lab with state licenses. We've received some grant support from the European and national governments in Germany. And the reason the FDA logo here is because we're also a registered medical device manufacturer as well as being a laboratory, which means that in addition to having laboratory activities, we also manufacture the devices that are used in the ALCAT test and the reagents that are used in the ALCAT test and the other cellular functional tests for micronutrient assessment. So that adds an extra layer of regulation to what we do, and so it helps with the quality. So to get into the discussion, I'd first like to give a little context here. This is a picture of the cover of Science magazine, a very well-known journal, old journal, from 2013, quite a while ago, where there was a special issue dedicated to inflammation. And what you see here is a human lymphocyte, and superimposed upon it is a yin-yang sign. And the whole theme of the issue was that there was a need for resolution of inflammation, as well as proper function of the immune system. And we sometimes forget that. We tend to think of this immune system as a bunch of warriors that go out and protect us and fight. But just as important is the yin part of the immune system, which resolves tissue damage following inflammatory attacks or invasions. And there's a little bit of yin inside the yang, there's a little bit of yang inside the yin, and it has to stay in balance. And for us, that means balance between the innate and adaptive branches of the immune system, as well as activation and repair. And that's what we're going to talk about today, essentially, is use of diagnostics and how they can be applied in terms of naturally achieving this balance. Main points are the chronic activation of innate immunity underlies degenerative disease, as well as suppression of specific immunity. And that's a key point. The things that trigger the immune system may be pathogens, and they activate receptors called PAMPs, which is an acronym for pathogen-associated molecular patterns, or they could be sterile triggers like foods and chemicals, or even cellular debris like cell-free DNA. And they're called DAMPs. They're similar, but they recognize danger-associated molecular patterns. Cell-free DNA is an area of active research. There's about 4,000 references on PubMed. We're going to discuss that, and that's a major DAMP. And we have developed some functional cellular response tests that can assess immune health, status and immune function, and can predict. The new testing is aimed at predicting morbidity and death. And we'll talk about this inflammaging markers. We can identify sterile triggers of inflammation to help achieve the balance we're seeking through ALCAT testing, as well as micronutrient testing through the CNA, which is a cellular nutrition assay, which is also a functional cellular response to micronutrients and antioxidants. And we'll discuss that in a little bit more detail. And I want to emphasize at the outset, because we're going to get into this, it's more important now than ever. that we look at healthy immunity, and I think everybody recognizes that. So one of the things that has been shown to us through our experience journeying through this COVID experience is that females have a better outcome than men. They have a 40% greater chance to survive than males if they have serious disease. The neonates, infants, newborns, also, if they have issues, females are much more likely, they have a greater advantage to survive infection and inflammation. And the aim here of this study was to quantify expression of toll-like receptor 4. And the reason for that, and the reason why females have a better chance of surviving COVID and illness at birth, is because of the fact that all these toll-like receptors are encoded on the X chromosome. And girls have 2 of them and boys have 1 of them. So girls have more toll-like receptors. Now that could be a good thing, couldn't it? And that's part of the balance of the immune system, is to protect us. But there can also be a time when it's not such a good thing. For example, just last month there was a study in Nature talking about toll-like receptor 7 and how if in the case of this situation where they found a small variant in the toll-like receptor 7 gene, again, expressed on the X chromosome, which females have 2 of and males have 1 of, there was a greater likelihood of developing lupus. In fact, people with that, or in this case, the child where they identified that had severe lupus. And when they implanted that gene into mice, they found that they would induce lupus. So it brings home, it's a double-edged sword. So the females have the advantage of greater protection, but also greater sensitivity. Now the pathway for this development of lupus was through the innate immune system, through this toll-like receptor. And why is that? Because this receptor recognizes cell-free DNA, guanosine, which is one of the nucleotides that we find in the genes of bacteria, but it can also come as a dam from our own damaged cells. And that's what we want to develop. Now, so that's a little bit of background in terms of the first stage of how we protect ourselves when COVID arrives is the innate immune system, and the females have an advantage there, and the males have a disadvantage. What else is involved in COVID? A paper from just a couple of years ago, when COVID was first developing, talked about the ACE2 receptors being much more than just a receptor for SARS-CoV-2. And the ACE2 receptor is very important. It's expressed on certainly epithelial cells in the lung, but also it's distributed widely throughout the body into the kidneys, into the brain, and various other tissues which are affected by COVID. So when we have a situation in which the receptors are blocked because they're blocked by the spike protein, not only are we having the invasion and the infection with the virus through the entrance of the ACE2 receptor, we're also losing function of those ACE2 receptors, which then leaves us prone towards the lack of protective benefits that the ACE2 receptor activation confers. Now, an interesting paper talking about the vaccine, because getting the vaccine is almost like getting the infection, and that's the intention. That's always the case, not just with COVID, but purpose of infection is to give a weaker dose of an attenuated virus or something in this case, which is a brand new approach. And we want to look at that for a second and consider that the name Moderna means modified RNA. Now, this paper coming from 2008, got the papers mixed up. This is a review just last year of why Moderna and Pfizer vaccines have a Moderna means modified RNA. So the uridine nucleoside in the RNA has now been synthesized. And the advantage of that is that the synthetic uridine can actually not degrade and find its way into the cells more easily. But that's based on research going back a long time that we don't necessarily hear about in the context of the vaccines today. That the modified pseudouridines have a higher translational capacity than the natural RNA. And the encoded protein can be detected in the spleen at 1 and 4 and 24 hours after injection, which is kind of interesting because the understanding at the outset of the use of these emergency authorization of these new vaccines was that they would degrade pretty quickly. They'd be injected into the muscle tissue in the shoulder. They would teach some cells how to translate that RNA into the protein of interest, which of course is the spike protein. And they would not last for a really long time. However, what we see going back to 2008 is that it was well known that this synthetic uridine would last a long time in the body. And also it would escape, it was not immunogenic, it would escape the immune system's degradation of it. And the conclusion of this paper was that improved properties conferred by pseudouridine make such mRNA a promising tool for both gene replacement and vaccination. The reason I mention that is because what we're finding out is that people have long COVID. And Dave Brady's going to speak about that in more detail following my presentation. But that kind of challenge, which indicates that there may be a persistence of this foreign RNA in our system and reaching into our tissues for much longer than we anticipate, is all the more reason that we need to have a healthy immune system that doesn't overreact to it. So the other troubling thing about it is that patients often remain positive for viral RNA as detected by PCR many weeks after the initial infection. And that the RNA from the COVID, SARS-CoV-2, can be reverse transcribed and integrated into the genome of the infected cell. So this fits in with what we were just talking about, that such chimeric transcripts are detected in patient-derived tissues, in the very same tissues where the ACE2 receptors, which protect us, which teach the cells to protect us, are downregulated. There's a lot of papers. This one just came out a couple of months ago in a special issue of Science Journal of Experimental Medicine, talking about the need to understand the immunopathology of COVID. Because of the immune response to COVID is a key determinant of severity and outcome. Understanding the immunological underpinnings of COVID pathogenesis is critical to predict, prevent, and treat infections, to prepare for the possibility of future infections caused by similar coronaviruses that may be introduced from existing reservoirs. And so they decided to look at the immunological underpinnings of the pathology from different perspectives over time. What they found by using a number of different technologies, it was a multi-omic approach, was that in non-severe COVID, there was a lot of neutralizing antibodies and a strong T cell response. Now, the T cell response, it's important to understand, is what we really want. It's not really talked about so much. It's beginning to get talked about. Well, we need to understand it more because people talk a lot about the antibodies. Well, you have the antibodies, that's good. But the antibodies are intended to neutralize it to prevent the virus particles from binding to the cells. But when they do, and there seem to be enough breakthrough cases around to understand that they do happen, we need T cell defenses. And T cells are cells of the adaptive or specific immune response. And a strong T cell response is associated with a successful outcome or less severe disease. So patients with mild COVID have much lower levels of pro-inflammatory plasma cytokines and higher levels of tissue repair factor, suggesting that the immune response in mild disease can work to stave off the virus and without triggering hyperinflammatory states, which we commonly call the cytokine storm, which usually is what kills the person. Severity-associated changes in neutrophil phenotype included upregulation of pro-inflammatory cytokine encoding genes. So neutrophils are innate immune cells. And of course, as you've probably learned earlier today, or you know from basic immunology studies, that neutrophils are the major component of the innate immune cell population. They are about 70% of leukocytes overall, and they are short-lived, and they are not specific as the adaptive T cells and B cells are. And they produce a very aggressive first line of defense that, if it's successful, will then prevent us progressing to a need for the adaptive immune response. But there's a downside to that, as we're going to explain shortly. So upregulation of several epigenetic regulators involved in neutrophil activation, like PADI4, which is required for formation of nets or neutrophil extension traps. That's, again, we're talking about severe cases. So severe COVID is associated with a number of distinct immunological signatures, such as increased levels of pro-inflammatory cytokines, such as IL-1B, interleukin-6, and IL-10 and IP-10 and TNF-alpha. Those are associated with worse outcomes. And that's important to understand that those we'll explain, give the context to that later on. But for now, we want to just understand that we want to have a more robust T cell response and a balanced and not so hyper-aggressive innate immune response. If we just keep that in mind, everything else that comes here in this presentation will make more sense. So we've developed a couple of new tests to determine a person's immune system preparedness to deal with infections like COVID, as well as the general challenges that we experience through life, because we move through an environment where we actually have to defend ourselves millions of times a day. So our tests, our new tests, consist of 4 different components. And the first one is measurement of the activation of an immunological pathway called JAK-STAT, which is an acronym for Janus Kinase Signal Transduction and Activator of Translation. And what we do is we make a measurement of fluorescence intensity in 2 different classes of leukocytes, monocytes, which are innate immune cells, and lymphocytes, which are adaptive immune cells. When we stimulate them, we activate them with 2 different cytokines, interleukin-6 and TNF-alpha, which are produced by innate immune cells and other cells to activate the immune system. So when we trigger the immune system like this artificially, we want to see that it actually can turn on. And I'll explain how we do that. We use flow cytometry to make that measurement. The second part of the inflammation panel is quantification of inflammatory cytokines in the serum. IL-1 beta, TNF-alpha, IL-6, the usual suspects in poor outcomes, and things that trigger a lot of things we don't want, like metabolic syndrome and various chronic inflammatory conditions. CXCL9, which is produced by senescent endothelial cells, which is probably the best marker for predicting cardiovascular disease and also mortality. And a couple of others that, if we have time, we'll discuss. And then a simple measurement of the ratio of neutrophils to lymphocytes. Again, innate immune cells to adaptive immune cells. And an optional component, which is the telomere length, the average telomere length in leukocytes measured by quantitative PCR. So we took a lot of the direction from stuff in the literature that we found to be pretty important. This paper from a few years ago called Defective Signaling in the JAK-STAT Pathway tracks with chronic inflammation and cardiovascular risk in aging humans, and published in Cell. And super strong predictor of how well a person's overall health is reflected in their ability to harmonize with the environment. We focus on the STAT signaling pathway because it's sensitive to a number of different inflammatory mediators, such as those found in higher concentrations in the circulation of aging individuals. So the JAK-STAT signaling pathway in immune cells plays a critical role in regulation of immune responses. Abnormalities are associated with immune disorders, and the pathways is regulated at various steps involving crosstalk with various cytokines. Contains various forward and backward feedback loops. And essentially, it would look something like this. We're looking here in this schematic of the cell membrane without the stimulation, and then stimulation of the cytokine to the Janus kinase receptor, which then sends signals through the pathway, which ultimately pass along these signals to enter into the nucleus and attach to the promoter region of genes and turn on genes that produce inflammatory mediators to undertake an immune response. This is what we want to see when we artificially stimulate this pathway. And the process involves phosphorylation. And phosphorus molecules come from the ATP produced in the mitochondria through normal aerobic metabolism. So you can see the important link between mitochondria, this is very one of them, and proper immune system function. And we measure this using flow cytometry. Now, flow cytometry, simply put, and forgive me for going through these slides pretty fast, but we're going to have time constraint here, is a system where we're measuring any molecule inside the cells or on the surface of the cells to tell us what kind of a cell it is, or inside to tell us, for example, if the STAT135, in our case, stages of activation are very active or not. And we do this by attaching antibodies that are specific to each one of the molecules that we're trying to target. And we flow that through a laser beam. And the hydraulics are very sophisticated. There's different streams of liquid going at different velocities to focus the stream of containing the cells so that we have very tight and narrow alignment there. And we shine a laser light on it. Laser, of course, means light amplification through stimulating emission radiation, which means that all the light waves are there of a single frequency. So we have coherent light. And then when that light is refracted and goes through various filters, it will generate the signals that are produced when the laser light interacts with the fluorophores or dyes that are attached to the antibodies that are specific to the molecule in question. So that's a quick, usually takes several hours to explain flow cytometry, so don't worry if you don't remember all of it. But the idea is that we're able to detect activation of molecules by looking at phosphorylation of the targets STAT1, 3 and 5 through this technology. And as it goes through different filters, the different emissions from the different fluoroforms are detected and by photomultiplier tubes, and that's converted to an electronic signal that then is recorded in our data software. And so from that, we're able to determine, yes, we stimulated the cells with interleukin-6, tumor necrosis factor alpha. We want to see a healthy immune response. This is how we can determine with quite a bit of accuracy if we're seeing what we should be seeing. And the results of investigations of correlating these things with clinical outcomes is that cytokine signaling response of older adults is reduced with aging, regardless of how much of a stimulus you try. Reduced responses are correlated with increased baseline levels of T-stat, which is phosphorylated stat, and also elevation of memory cells. What that means is that your immune system, or the person's immune system as they get older, or if they have chronic activation, they're depleting their resources. They're being activated inappropriately when they should be resting. And then when the proper stimulus comes along, there's less resource to fight in that situation, to fight the infection. So again, an imbalance in the immune system, an imbalance between yin and yang. Too much yang, not enough yin at baseline. So that happens when we get older, but of course we know that if we live a proper healthy lifestyle, we can delay that and have a much longer health span. And this does give us an indication into that. So reduced responses to cytokine stimulation are also due to elevated basal levels. And then we observe differences in naive and memory cell responses. What that means is that T cells that have more memory, or in a way more, it's good to have some memory cells, but too many and not enough naive cells means we're not looking at the environment in a fresh way. We're looking at it in a biased way from our past. Reduced signal responses are associated with atherosclerotic burden and diastolic dysfunction. And as was mentioned, CD4 positive, these are different types of, these are helper T cells, and these are cytotoxic T lymphocytes, but they're lacking the ability to recognize new antigens. And that's a hallmark of immunosenescence. So we want to improve that situation. And just briefly, what these STAT proteins are associated with, different immune functions. The other part of the inflammation profile includes the cytokine evaluation. As I mentioned, ones that were familiar with IL-6, TNF-alpha, interleukin-1 beta, we know cause chronic inflammation. CXCL9 is a key one that we wanted to include because it is probably the best predictor of cardiovascular disease that one can do. These others, like CXCL2 and CXC2, are associated with various normal functions. We don't want to see them too high. Elephant is associated with inhibiting too much elastase. And some of these were selected to be included in the panel from research we did in collaboration with folks at Yale, where we looked at clinical outcomes in response of a therapeutic LCAT diet. And the investigators, one of whom was not only a gastroenterologist, but he had a doctorate in immunology, had the forethought to bank the patient's or the subject's serum at the outset of the study so that we can retrospectively go back and look at the people who had the best clinical outcomes and see how their cytokine profiles changed to see what's most important. And these are key ones that we found there. So that's a little bit of background on testing and a little bit of background on the role of those cytokines specifically, short overview. But now we want to talk in a little bit more detail about the mechanisms by which people have worse outcomes from COVID. And just in January in Science Immunology, another one of the science journals, which is it's a new journal, it's excellent, found in this paper, there was a report of the finding that there were increased neutrophil extracellular traps. Remember, it's overactivation of the innate immune system, one mechanism of which is neutrophil extracellular traps are found in the lungs of patients with critical COVID. And that also has been shown to induce death of epithelial cells and contribute to immunothrombosis. Now, these cells also will suppress T cells, which T cells, for one thing, produce interferon gamma, which we need to kill the viruses. And looking at a normal case or a healthy outcome, we see there's not much going on. We have an alveolar in the alveoli, macrophage, which is there to deal with cleanup of any problem that happens. We have an normal exchange of gases between capillaries that are nearby. In mild COVID, we have, again, these macrophages get active and produce interferon, dendritic cells get involved, and we have some neutrophil activity producing these cytokines that we talked about. Critical COVID, we see a more pronounced situation, fluid coming in because the barrier function in the alveoli has been compromised. Neutrophils are getting activated, producing free radicals and traps and fibrin and causing the depletion or the exhaustion of lymphocytes. So once again, not to be like a broken record, but the important thing here is to have the balance between the innate and the specific immune system. And it's the innate immune system which is associated with the worst activation of the innate immune system to a hyper degree is what causes the serious complications. The neutrophil lymphocyte ratio. So this paper talks about neutrophil to lymphocyte ratio in relation to all-cause mortality. And poor immunity is related to all-cause mortality and cardiovascular events. So again, we want an immune system that can respond, but is resting when it should be resting, not reacting to a whole bunch of environmental triggers inappropriately. So in this meta-analysis, 8 studies, it was shown that the people with in the highest quartile of the neutrophil lymphocyte ratio had a 2.33 times more likelihood of dying than people who were in the lowest quartile, and almost a double risk of having cardiovascular events. So the meta-analysis indicates that NLR is a predictor of all-cause mortality and cardiovascular events. That's why it's a very simple test, and we included it into the information panel. I'm not going to spend too much time on this, but this was a study looking at older subjects, published just last year in Nature Aging, another Nature journal. And again, we see that inflammation, low-grade systemic inflammation in older humans, termed inflammation, is characterized by higher serum levels of inflammatory cytokines, IL-6, IL-1 beta, TNF-alpha, and C-reactive protein. But we measure only IL-6 because that induces C-reactive protein. This elevated expression of inflammatory proteins is a strong predictor for frailty and mortality. Mechanisms that contribute to inflammation are multifactorial and include increased numbers of senescent cells. This is one of the things that telomere tests can tell us, but also when telomeres get too short, the cells can no longer divide, they become senescent. Also increased gut permeability, not a surprise to anybody listening, and expression of DAMPs, such as cell-free DNA, and failure of the immune system to resolve it. Now, another paper I just found interesting, I wanted to pull this quote from, is that Western diet triggers and this receptor, NLRP3, nucleotide-like oligo receptor protein 3, is inside the cytosol. It's a little bit different from the toll-like receptors, but it's also an innate immune cell activator. And these authors in Cell, the journal Cell, write that it's conceivable that sterile inflammatory triggers, such as a Western diet, can induce trained immunity. So the innate immune cells undergo training, which is mediated by epigenetic factors, as opposed to adaptive immune cells, which can actually change their genes to recognize particular pathogens. And in such a scenario, a secondary stimulus would then trigger altered and potentially pathological immune responses, which shows, again, too much yang. We have ongoing chronic activation of innate immunity, which makes the immune system more prone to be overactive. Then something like COVID comes around and we have a cytokine storm. And that's very simplistic, but essentially that's what we're talking about without all the details. So we want to, again, balance less yang, in this case more yang. The Nature Aging Journal recently, last year, we took a lot of inspiration from this research done at Stanford, looking at the JAK-STAT and the cytokines, describe their observations that chronic and systemic inflammation is thought to be triggered by physical, chemical, and metabolic stimuli. So again, sterile agents, not necessarily pathogens. And in our environment, there's more of that. And such as those released by damaged cells, DAMPs. This inflammation is associated with aging. Is characterized by low-grade and persistent inflammation. Ultimately leads to tissue damage in various organs. Now, just to put it into context, this was a paper that came out before all this understanding about COVID. Earlier on at the time when we didn't know as much about the cell-free DNA as we know now. But these people from the UK made the statement that their belief is that the current knowledge on cell-free circulating mitochondrial DNA is in a rather exploratory phase with the potential for the future to rewrite the pathology of the leading causes of morbidity and mortality, autoimmune disease, stroke, and so forth. This is what a net looks like. This is a neutrophil that has undergone netosis, and you see things that look like nets that go out and trap pathogens, and it's comprised of the membrane structure melded together with inflammatory mediators from the granules inside the cell, as well as the chromatin, which means the histone and the DNA that's wrapped around the chromatin in the nucleosome and the nucleus. And the modified histone proteins and the DNA themselves, when they get released, are toxic to these microorganisms and that they get trapped. And that's the beauty of this thing. That's the good part. But they're also toxic to our own tissue. And if we have too much of this going on and we don't have enough of the enzymes to break this down, then we get autoimmune disease as well as other damage to tissues and organs throughout the body. Now, I haven't updated this for the last 6 months, but these are publications on PubMed having to do with neutrophil extracellular traps. And you can see inflammation 1200, COVID 55, and it's more now, COVID fatality 14, thrombosis, cardiovascular 700, autoimmunity 470, aging, dementia, respiratory disease, pneumonia, eye disease, liver disease 563, and counting. So it's becoming well understood. And I think what these people said back in 2017 that we're maybe, I don't know if we'll rewrite all the pathology textbooks, but we're going to rewrite a few chapters for sure. Immunothrombotic dysregulation in COVID-19 is associated with respiratory failure and coagulopathy, microvascular thrombopresence in lung, kidneys, and heart. Just the organs where we block that the ACE2 receptors are expressed in. The next, another paper, again, before COVID, talking about the dark side of neutrophils. Again, I don't know if it's yin or yang, but probably more in the yang. Nets contain antigens, some of which are modified histones, acting as neoantigens to induce formation of autoantibodies, induced to accelerate vasculitis, rheumatoid arthritis, and thrombus formation. The nets spur formation of autoantibodies and may also serve as a scaffold for thrombosis, providing a link among infection, autoimmunity, and thrombosis. And COVID is a thrombotic disease. Just briefly, this was a summary of the clinical collaboration we did with Yale, which led to the proteinomic analysis. And essentially, they took patients who had met the criteria of IBS and certain other criteria that they were not taking drugs at the time and had surgeries and so forth. Did an LCAT test at baseline, put the control group on the LCAT real diet. The control group went on a placebo or sham diet, also based on LCAT, but leaving in the test positive foods and looking at outcomes over 4 and 8 weeks. And there was a major change in the major difference between the two groups. And as I mentioned, they did a proteinomic screen. And one of the findings was that there was a higher level of plasma elastase in the people before they went on therapeutic diet, and it dropped dramatically. So elastase is why we have elephant in our panel. Now, elastase will also cause intestinal permeability, as well as damage to tissues throughout the whole body. So it makes a lot of sense. And here's just a schematic showing what they found in a follow-up study, again, with LCAT, the same group from Yale, using an LCAT positive food versus an LCAT negative food. An LCAT positive food via protein kinase C would cause the release of both nuclear and mitochondrial DNA, which to leave the cell and cause the things that we talked about. Following that, there was another study done at Georgetown and University of Leipzig with our group, trying to determine whether the DNA was from the mitochondria or from the nucleus. And unfortunately, we don't have the time today to get into too much on that. Maybe in the Q&A we'll have some time. But of course, mitochondria resemble bacteria. And so mitochondrial DNA right away off the bat is pro-inflammatory. But nuclear DNA is also dangerous because it contains these histones, which are toxic. So we found that, again, there was both. When you had an LCAT positive food, there was more cell-free DNA, not so with an LCAT negative food. So we do think an LCAT test can predict what foods will cause release of cell-free DNA. So how does it work? It's a cellular test. And what we do is an ex vivo challenge of patients' whole blood while the cells are still alive. And the analysis also uses flow cytometry, but it's called impedance-based flow cytometry, where the cells flow through a sensing zone and they're suspended in an electrolytic solution, and there's an electrical current going by. And the cells will impede the electrical current in proportion to their volume. And so by measuring the cells one at a time and aggregating that measurement, we get a size distribution curve. Now, we do a number of samples that don't have a food allergen or a chemical allergen or an herb or whatever we're testing for. And That's the patient sample coming in the door as the patient is. And we average those together and get a baseline curve. And then we do the same measurement and generate a size distribution curve of the white cell population for each individual test sample, and then we compare them. And if there's a positive reaction, the cells either get bigger or degranulate. And then that can lead to a number of different things, release of mediators like elastase and myeloperoxidase, and free radicals for sure, and also cell-free DNA. So it's a functional cellular response test in real time. It's not based on some theory that an antibody might do something that might cause a reaction. It's your cells are reacting, and these are the cells that mediate disease. So it's kind of simplistic, and that's why when you compare an ALCAT test with actually a feeding challenge in experimental conditions that are tight and rigorous and blinded, you get a high correlation. So you only get that with ALCAT. It's been attempted with others and it doesn't happen. So talking again about the DNA, it's not just encoding our proteins. In fact, only 1% of the DNA actually is used for encoding proteins. The rest of it is epigenetically controlling the introns and determining what gets expressed. And if you 100 billion neutrophils, all containing lots of DNA, die every day normally. And if you stretch it out, it would reach from one end of the solar system to the other. And there's like a little over 6 feet of DNA coiled up inside the genome of each cell. And unfortunately, just due to time restraints, I wanted to get into a little philosophy here I think is interesting, but we'll do that if there's time for the questions. But just briefly, I started studying Vedic philosophy a little more than 50 years ago, and I was interested to see kind of parallels between Vedic philosophy, just as it relates to our understanding or their understanding of karma, which is very similar to genomics and then epigenomics and modifications to genomics through lifestyle. I'm sorry, I don't think we have time today, but if there is time at the end, we'll definitely talk about that. So after all these 100 billion cells die normally, never mind the cells if we have an infection, then there's more. They get opsonized, they get cleared. That has an impact on further inflammation. And these cells that undergo this apoptosis, which is this normal suicidal thing to happen when the cell's time has come, emit find me and eat me signals. Believe it or not, those are scientific terms. And then macrophages will then recognize those signals and then eat up, macrophages are big eaters, eat up those dead cells and clean house, which is very important. And they do that through these integrins that are on their surface that bind with those eat me and find me signals that are expressed on the apoptotic cells. And unfortunately, neutrophil elastase will inhibit that. The nets, and again, the neutrophil elastase is a big component of the nets, prevents the cells from being degraded. And this is a schematic that shows that. Of course, the scale is wrong, but macrophages are usually bigger. They would bind with the apoptotic cell and digest it, clean house. But unfortunately, the neutrophil extracellular traps and the neutrophil elastase inhibit these molecules from performing their function. And over the years, there's been a number of studies showing how when you inhibit these processes through guided diet, elimination diet, modification based on ALCAT, a number of different conditions are improved to a great extent, some more than others. The test results, again, the degree of variance between the control curve and the the baseline curve are expressed as moderate or severe or borderline or okay. And one of the other things besides NETs that we want to consider are the free radicals that the body produces to neutralize pathogens. And one of the things that caught my attention several years ago in a book by Clark from UCLA was that the damage done by oxygen radicals extends to every part of the body, can attack and deform protein, disrupt structural complexes, and inhibit enzymatic functions, and also damage DNA. It also, when you have this inflammation, the body tries to quell it. There's more cortisol produced, and that suppresses the HPA, which then is not good, and it suppresses sex hormones. Excuse me. Inflammation attacks, shortens telomeres, and as telomeres get shortened prematurely, it's related to cardiovascular disease. We see that there's more oxidative stress and more neutrophils circulating in the nervous system of people suffering from dementia than from normals. MPO, another neutrophil mediator, and also other granulocytes produce it, is a better marker for cardiovascular disease because it tells us about the oxidation of LDL, which then becomes toxic and forms, helps form plaque. And that oxygen radicals play a role in preeclampsia and also in male infertility. We see higher levels of macrophages and neutrophils in the semen and more reactive oxygen species there too. So that's that part of how to remediate and improve the immune system function by quelling chronic inflammation of the innate immune system. We also want to look at supporting the adaptive immune system. And for that, we use a test that, again, is a functional cellular test where we, well, it's based on a study done in the 1940s that had won the Nobel Prize in 1958. And this research was carried out at University of Chicago by Beadle and Tatum. And they actually did this study where they took the spores of a common bread mold and found that they can grow these, they would replicate in an essential growth medium just fine until they irradiated them. And then in order to see if they can restore function, they added back individual nutrients to this essential growth medium one at a time and observed to see if they would restore growth of the cells. After 236 times, they found that arginine would restore growth of the cells. Kind of like with MTHFR, if there's a deficiency in that enzyme pathway and the other genes in that pathway, you can treat it by adding back things, B vitamins, for example. We also do that test as well. And why is that important? Because the specific immune cells, this is an old graphic from Scientific America, but it shows when a pathogen is presented to a T cell. And again, it's only a T cell that has a receptor that recognizes that pathogen. It then multiplies and then becomes activated and then sends signals to B cells that recognize that pathogen as well. They multiply, convert to plasma cells, which secrete soluble forms of the receptors that recognize the pathogen, and we fight the infection. And it all depends on the ability of the cells to multiply. And this is required, this requires nutrients. So if we, as we do in our test, the cellular nutrition assay, we separate the lymphocytes, add back the patient's serum so we have the kind of situation that's the cells are used to. We stimulate with a mitogen. It's not a pathogen, but a mitogen, a mitosis generator, artificially. So we want, this is intended to stimulate division of all the cells. So they should proliferate if they have enough nutrients within them. We then add micronutrients one at a time, and then we compare the growth rate of the aliquot where we've added the micronutrient to the baseline. If we see enhanced cell proliferation, which is desirable, we can infer that there's suboptimal levels of that micronutrient inside the cell for whatever reason. So this is only one of two cellular tests on the market for micronutrients. And you can choose between them. We report out deficiencies in the red versus green, and we test about 48 substances currently. We also challenge the cells in a separate assay to look at how they resist challenge with hydrogen peroxide. And then we also do those challenges and compare that to a group of normals. So we see how a person sit as to how well protected they are. And then we add back antioxidants into the serum while we do that challenge to see which ones protect them the most. And then we report it out as being either sufficient or could be benefiting. So putting it all together, you could take all these tests and first assess the immune status and health of the immune system by using the assays we described. The JAK-STAT assays and the serum cytokines and chemokines and the neutrophil lymphocyte ratio, all in one test, telomeres if one wants. Then identify through ALCAT which environmental substances, foods, chemicals, herbs, molds, drugs are causing chronic activation of the innate immune system. Change the diet accordingly, run the cellular nutrition assay, add in the specific micronutrients that improve the function of the adaptive immune system and protect those cells from free radical damage. And then repeat, reassess, and adjust periodically.
Roger, we are out of time. But great. Here's your contact information. So if people have additional questions, they can go to your website. With that, I really want to thank Roger for a wonderful presentation, a lot of great information. So thank you for your time today.
My pleasure.